Thanks for downloading a 3CR podcast. 3CR is an independent community radio station based in Melbourne, Australia. We need your financial support to keep going. Go to www.3cr.org.au for more information and to donate online. Now stay tuned for your 3CR podcast.
Hello and welcome to Lost in Science for another week. My name is Claire and as per usual, we are here to bring you some of, well, our favourite science of the week that we think you are going to enjoy just the same. And who are we? Well, I've got Stu here with me this week. Hi, Stu.
Hi, Claire.
Tell us about the science that's been tickling your fancy this week. What have you got for us, Stu?
Okay, look, I've got to admit this was one that I stumbled across earlier in the year and put it away in my get back to this file. And I finally got back to it. This is a paper that was published in February in Nature about how humans and different animals perceive time differently based on how they see the world, basically. So, and also how that's got to do with the world they see. So it's kind of a weird, timey-wimey feedback loop that creates different perceptions of time and different perceptions of the world based on the world that they're perceiving and the time they have.
Wow. This is going to be fascinating because I've thought about this a lot and then I just get stuck at how do you measure that and how do you quantify another animal's experience?
We may be able to quantify it. We may not be able to qualify it. So
yeah,
I'll explain that.
Oh, love that. Well, speaking of time and speaking of animals, I will be talking this week about someone who has been around for 100 years. I am, of course, speaking about David Attenborough, who's just celebrated his 100th anniversary, birthday anniversary, his 100th birthday, and has been such an incredible inspiration to millions and millions around the planet. But I wanted to bring it back to the animals because David Attenborough, as a naturalist, as a communicator, as someone who is incredibly inspiring, has a lot of animals named after him. Like not just a couple, but a lot. Yeah, over 50. So today I'm going to take us through a couple of my favorites. And you know, it's across the board. And I should say, I mean, I'm going to talk about the animals because I love talking about animals, but he has had things that living creatures that aren't animals named after him as well. I'm talking fungus, plants, protists. You know, he isn't selective in what he lends his name to.
He's the king of all kingdoms, really.
He is the king of all kingdoms, and happy birthday to him. So I'm really looking forward to celebrating David Attenborough in that way and just absolutely massacring some Latin names in the process, Stu.
Excellent.
On with the show.
Have you ever noticed how time seems to move at different speeds depending on what you're doing?
Yeah, yeah, and also what time of year it is. It's almost middle of the year. It's going pretty quick. But January always seems to take about 6 months.
And I don't know if you remember, but researchers in 2023 explained that, for example, during COVID lockdowns, people's perceptions of time were altered by the repetition and monotony, and they even introduced this term. I love this term, Blur's Day, to put a name to it.
Every day was Blur's Day, wasn't it?
Every day was Blur's Day, which is hilarious. But it's hard to pinpoint dates or times when every day feels the same and plays out the same. And that's also related to a slightly different phenomenon, which is connected to novelty, which is something that is much more perceptible as we age. But basically, time zips by when you're doing new things, and it drags when you're doing the same old, same old. In other words, time flies when you're having fun, and familiarity breeds contempt. I mean, it's no wonder that we have
it's a scientific fact.
Aphorisms have slipped into the language. But look, our perception of time is very much related to what we experience and a lot to do with what people see. But human vision is not the only kind of vision in the animal kingdom. So what we see has a lot of impact on our perception of our environment, but the human visual apparatus is very much linked to how we move around in the world and what we need to be aware of in the world that we move around in. So our vision is very clued into what historically humans have needed to know about their environment. But in the animal kingdom, vision's a much broader spectrum. There's so many different kinds of eyes, aside from the binocular lensed vision that human beings have, which we share with a lot of other animals. You know, we're not that dissimilar to birds and reptiles and mammals and all of these other animals that have the same kind of vision that we do. It's not just the kind of eyes we have, though, that is that goes into how we perceive the world around us. So let's just look at moving pictures for a second. Perfect topic for radio. But in that second of looking at a moving film image, we see around 24 individual images per second, more or less. And it's the brain that puts them together into a moving image that can be recognized, which is it's a trick that we've figured out to make images look like they're moving. Depending on how the image is viewed, it may not be apparent to other animals that there's even anything there to look at. It's not necessarily going to be something that will grab their attention like it does with people. People always will turn to a moving image. They just can't help it. But a static computer screen with a still image is actually flashing over 240 times per second.
Oh, wow.
But human eyes are not capable of registering flickering at that rate. So that's because the human eye only samples at a rate of about 65 flashes per second. So it's not fast enough or in sync enough to catch the flicker of an LED screen.
Right, but it is more than 24 frames a second.
It is, but that's, we could probably increase film footage to faster than that. But if you think about the extra energy and everything that's required, doing it would just be a waste of energy when we can trick ourselves that it's moving at 24 frames a second. But a passing insect would see a computer screen as more like a strobe light. So it'd just be flash, 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 flash. And a snail would see it as just a kind of blurry blob of a light source. It wouldn't actually know what was on the screen. It wouldn't matter if it was a static image or a moving image. A snail would not be able to tell what was going on at all. So some of that is to do with the focusing lenses in the human eyes. Obviously, that helps us to pinpoint particular things. But it also has to do with the sample rates of eyes. And some scientists from the University of Galway wanted to find out more about that. So they looked at published measures of time perception across 237 species of animals, from jellyfish to land-dwelling vertebrates and everything in between. The whole gamut of animal being. So how they did this, they were using electroretinograms, which is a cool way of saying they were recording the activity of the retina of the animal in response to flashes of light at increasing rates. So they'd flash a light, they'd go, yep, the retina responded. And then they'd increase the speed of the flashes and see at what point the retina stopped responding. At that point, it can't see them. They're too fast. They're too close together. So it's not responding to each flash. It's just perceiving light. And at that, they've reached the eye's sample rate. So this, of course, has a name, which is the maximum critical flicker fusion rate. Just rolls off the tongue, doesn't it?
Okay, great. Explain.
It's also called the flicker fusion threshold. It's also been called persistence of vision, but that term is not used so much anymore because it's also about getting, you get after images in your eyes and things like that. So it's also been used. So we talk about the flicker fusion rate or the flicker fusion threshold. So what they found was that animals varied hugely in their flicker fusion threshold, from dragonflies at one end, which can see about 300 flashes per second, or hertz, so we say 300 hertz, all the way down to the giant African snail, which can only see at 0.7 hertz. So it can't even perceive one flash per second. So in between, there's a big variation, not just between the kinds of animals. Some birds can see up to 138 hertz, but most don't see that fast. Humans are at 65. But other mammals like dogs are at 84 hertz. But rats are only at 47 hertz. So it's not.
And so, what do they hypothesize is the reason for this diversity across species?
It is basically what kind of lifestyle does that animal have, and what does it need to perceive in its environment. So what they found most interesting was that the flicker fusion threshold of an animal is related most closely with the animal's environmental niche. In other words, if an animal lives fast, it needs to see fast.
Right.
For example, a predatory fish that needs to catch its prey by visual means, or a prey fish that needs to evade a predator that's looking for it, needs to be able to see things really quickly and react really quickly. So you find that predatory animals have faster vision. They can see at a higher rate.
Yeah.
And prey animals, if they want to live for a long time, also have to see at a faster rate. But they also found that it varied depending how much light. So there's isopods that live on the ocean floor that there's so little light, there's not much point in them looking around at things. It's not going to help them all that much.
Did they also look at whether an animal was nocturnal or diurnal, awake during the day, and how much light's coming in and how much, does that affect?
It does make a difference, and that's why rats are at 47 hertz. It's lower than dogs. Dogs are awake in the day. Rats are in darkness, and move around in the darkness. But the biggest correlation was in how fast the animal moves. So, for example, insects being able to see at a much faster rate than humans is why they are often hard to swat. What seems to be moving so fast to our eye that it's a blur is a slow-moving bulldozer to a fly, because they're perceiving time at a much faster rate than us. It's like that guy in Austin Powers being run over by the steamroller. He could have just got out of the way, but he's just got slow in. But basically what they found, that an animal's vision and their time perception is shaped by the environment they live in. So it's not really surprising that there's a really tight correlation, and obviously a really strong selection pressure for those animals to see better, who need to see better in order to survive. And they have to, they adapt to the threats and benefits they need to negotiate in that environment. So it's a really interesting study, and it does go to show that even though we're all living at the same rate, objectively, subjectively, the speed of what we do is different depending what kind of animal you're actually addressing. But I thought it was it still doesn't really explain why shop-bought milk is the world's fastest drink, because it's pasteurized before you see it.
What are you onto? Anything of interest to the scientific community?
Together, you and I are going to make the greatest single contribution to science since the creation of fire.
It's a big scientific experiment. What do I know?
Across Australia on the Community Radio Network, you're listening to Lost in Science.
So, Stu, as you know, it was David Attenborough's 100th birthday recently.
Yeah. Does he have a fan club? They should have a fan club called the Attenboroughs.
Yeah. It is a good name, but I hate to say it, everyone on planet Earth will be a member. Because I've never met anyone who isn't a fan of David Attenborough. He's an inspiration. I mean, he's definitely one of my heroes. You know, he brings awe, wonder, respect for the entire natural world, and he has been doing since the 1950s. So today, you know, I want to mark the occasion because you don't normally get to mark the occasion of people's 100th birthday. You sort of, these sorts of, you know, celebrations of people's lives often, unfortunately, happen at the end of people's lives. So it's quite a joy to be able to speak about someone who's still alive and still doing what he loves doing while he's still with us, and be able to celebrate with him. Today I wanted to take a taxonomical look at David Attenborough, because, you know, when you inspire as many people as he does, it's only natural that you get a few animals named after you. And he has had around 50 different animals named after him.
50.
It's
one every 2 years, but
I know, right? It is quite a lot, and the diversity. Sorry, I should go back and say 50 different species, because they, as I said in the intro, there are plants, there's fungus, there's protists, there's all types, and I'm being animal centric here, because I am going to talk about the animals, because that's why I watched David Attenborough. So sorry, Stu.
Hey, look.
You'll have to look up the fungus without.
The Private Life of Plants is worth watching, but it does have
it is a good one, isn't it?
It's probably the most exciting documentary about plants ever, because they just, you know, they still don't move around that much.
So let's get into it. So I want to talk about the very first species named after Attenborough, and it was actually an Australian species, the Australian velvet gecko, Odura attenboroughi. It was named in 1985. The Australian velvet gecko, it was later found to be the same species as another gecko, so the ocellated velvet gecko. So what happens in taxonomy when it's like, oh, we've named two things, but they're actually the same species, is I think you take the older.
Yeah, the one that was
species
named first gets precedence over the incorrectly discovered one.
Yeah, but it's still historically important because it was the first attempt to honour him. So we'll take that on notice, David Attenborough, Odura attenboroughi, and then go to what I think is probably the least glamorous tribute, and it is a tiny little weevil. A weevil being like a tiny.
Yeah, they're kind of beetly.
And it is
things.
beetly looking things with sort of like big antennae.
I think they have a big nose,
but they're also incredibly small. Oh yeah, a big nose. Yeah. Trigonopterus attenboroughi. It's tiny. It's flightless. It's Indonesian, and it's a weevil. And it's only 2.6 millimeters long. Now, this one's pretty unglamorous as well, because it was written up and described in a paper that described 98 different species.
So it's barely a footnote in that paper, really. Exactly.
And the lead scientist on the paper was like, I really like David Attenborough. And, you know, I'd run out of things in my room to name everything else. I was naming 98 different species. Yeah. So sometimes it's the tiny little beetles in the leaf litter that David Attenborough gets his name from. So from the tiny to the sublime, David Attenborough has also got his very own plesiosaur genus. So this isn't just a species. This is a genus. So this is a group of different species. And a plesiosaur, Stu, as you would know, it's not a dinosaur. It's like you water prehistoric species, right? Like
Yeah.
with the long necks, they sort of get mistaken for
Yes.
dinosaurs a lot, don't they?
Yeah.
It's a marine reptile, prehistoric marine reptile, I guess you would say. They don't exist anymore.
Well, you tell that to the people of
ballpark.
Loch Ness.
True. This one, which was Attenboroughsaurus coneyberry, was around 200 million years ago. Big long neck, flippers, like very classic plesiosaur. Think Nessie. And it was originally classified in the genus Plesiosaurus and then reclassified in 1993 to be Attenboroughsaurus, its totally own genus. And named for David Attenborough because apparently he loved, no surprises, he loved collecting fossils as a child. And discovered in Charmouth in Dorset in England. The original fossil was destroyed during World War II in the bombing of the Bristol Museum, but there's a plaster cast at London's Natural History Museum that's now the type specimen for this particular species.
Genus.
So close to home for David.
Yes.
Oh, sorry.
So we don't
genus.
really know how many species might have been in that genus either, because it was, we've only found some
of fossil.
Some of one. So yeah.
Yeah, exactly. Next, I wanted to talk about a fantastical one. This is another Australian one. It is a semi-slug. A semi-slug with an amazing colour scheme. It's another one that's been named for its genus, Attenboroughrian rubicundus. It's bright green and red. It's from Tasmania, and it's called a semi-slug because its shell is too small to retreat into. So it has a shell, but it can't. That'd just be
kind of annoying. It's like a jacket you can't put on. You just got to carry it everywhere with you.
It's like the haute couture of the slug world. It's like, a jacket that does
a non-functional
it's
accessory.
Just there for show. Exactly. Oh boy. Anyway, originally classified in another genus and then got its own genus of Attenboroughrian in 2007. We've also got a fossil fish that is not just any sort of fossil fish. This one changed vertebrate history. This is the Metapiscus attenborough, and it is a 380 million year old fossil fish from the Gogo Formation, another Western Australian one, and it is the first fossil preserved with an embryo and an umbilical cord. So they think it's the earliest known vertebrate shown to give birth to live young. I know. How cool is that? And the scientific name, Metapiscus attenborough, roughly translates to Attenborough's motherfish.
Of course, motherfish. Yeah.
What a great name. Motherfish. So it originally honoured because Attenborough had highlighted this fossil in one of his documentaries. So a great example of him just being awesome as a science communicator and increasing public awareness of, fossils and important fossil sites. And now for his 100th birthday, people wanted to do something special for him. So what do you do for the man who has his name on every species? Well, you get him a parasitic wasp. And that's what David Attenborough got for his 100th birthday. He got himself his own parasitic wasp.
So I tell you what, the species name will be. It'll be Attenborough.
Yeah. It'll be Attenborough. Yeah. Yep.
That's just how it goes when you're naming the species.
It's just how it goes, isn't it? Attenborough. So it feels appropriate for him. It's not cuddly, it's not charismatic, but parasitic wasps are incredibly important and fitting, as you know, he's championed all types of biodiversity across his career. Now, before I finish up, I do want to throw in a bonus here for you, Stu, which is not a species at all, but a ship that he was named after, which is the RRS Sir David Attenborough, which is a British polar research vessel. And I don't know if you remember back to the world's most famous, globally famous ship that was publicly named. Before it was the RSS David Attenborough, it was
Bodie
do you remember?
McBoatface?
It was Bodie McBoatface. So David Attenborough has also done all of us a public service by removing Bodie McBoatface and putting his name on it. So we're all thankful for that. Thank you, David Attenborough. All 50 species. Incredible to be named after so many different types of diversity of life on Earth. And, you know, I think one of the most touching parts is that, you know, many of the biologists, they're naming him not because he's discovered something, but because, you know, just like me and the millions of us around the world in the Attenboroughs, he's inspired them to look closer at the natural world in the first place.
Thanks, Dave.
And that's all we have time for on this episode of Lost in Science. Lost in Science is recorded on the lands of the Kulin Nations and the lands of the Ngunnawal and Ngambri people and broadcast across Australia on the Community Radio Network with the support of the Community Broadcasting Foundation. If you'd like to get in touch with us, you can find us at lostinsight@gmail.com or just listen again next week when Lyndon, Claire, Chris and Stu get lost in science.
Thanks for listening to a 3CR podcast. 3CR is an independent community radio station based in Melbourne, Australia. We need your financial support to keep going. Go to www.3cr.org.au for more information and to donate online.